Chapter Introduction. Molecular Basis of Inheritance. Background recap. In the previous chapter you studied how traits are inherited from parents to offspring, what we call inheritance patterns. Mendel called the controlling units of inheritance as factors, but did not know what those factors were made of. Scientific progress. Over 100 years of research after Mendel led to a major discovery. These factors are genes, and they are made of DNA, deoxyribonucleic acid. This discovery changed the entire course of biology, leading to the field we now call molecular genetics. What are nucleic acids? Living organisms have two types of nucleic acids: DNA, deoxyribonucleic acid; RNA, ribonucleic acid. You learned in class 11 that these are polymers made up of nucleotide monomers. Functions of DNA and RNA. Molecule main function, additional roles. DNA function as genetic material in most organisms, and stores hereditary information in the form of DNA sequence. RNA function as mainly a messenger, mRNA, which can also act as adapter, tRNA, structural, rRNA, or catalytic, ribozymes. In some viruses, RNA is the genetic material. What will you learn in this chapter? This chapter will help you understand structure of DNA, why it is called the blueprint of life, replication of DNA, how DNA copies itself before cell division, transcription, how RNA is formed using DNA as a template, genetic code, how nucleotide sequences in RNA decide the order of amino acids in proteins, translation, how proteins are made from RNA, regulation of gene expression, how cells control which genes are turned on or off, Human Genome Project, how scientists sequenced all human genes and what it means for us. What will you learn in this chapter? This chapter will help you understand structure of DNA, why it is called the blueprint of life, replication of DNA, how DNA copies itself before cell division, transcription, how RNA is formed using DNA as a template, genetic code, how nucleotide sequences in RNA decide the order of amino acids in proteins, translation, how proteins are made from RNA, regulation of gene expression, how cells control which genes are turned on or off, Human Genome Project, how scientists sequenced all human genes and what it means for us. 5.1 The DNA. What is DNA? DNA stands for deoxyribonucleic acid. It is a long-chain polymer made up of many deoxyribonucleotide units. Just like a necklace is made up of beads, DNA is made of nucleotides linked together. DNA length. How long is DNA? The length of a DNA molecule is measured in nucleotides or base pairs, BP. A nucleotide is a single building block. A base pair refers to a pair of complementary bases, AT or GC, in double-stranded DNA. Important point. DNA length is a species-specific characteristic. Different organisms have different DNA lengths. Examples of DNA lengths. Organism DNA length. Phi times 174, bacteriophage. 5,386 nucleotides, single-stranded. Lambda-phage 48,502 base pairs, BP. Escherichia coli E. coli. 4.6 times 10 BP. 4.6 million BP. Human haploid DNA. 3.3 times 10 BP. 3.3 billion BP. Why is this important? DNA is incredibly long, especially in complex organisms like humans. For example, if stretched out, the DNA from a single human cell would be about 2 meters long, but it's packed tightly into the tiny nucleus. This long structure needs a specific compact arrangement, discussed in upcoming sections like DNA packaging. Summary. DNA is a long polymer of deoxyribonucleotides. Length of DNA equals number of nucleotides/base pairs. DNA length is unique to each organism. This section sets the stage for learning the detailed structure of this long molecule in the next section. 5.1.1 Structure of polynucleotide chain. What is a polynucleotide chain? A polynucleotide chain, like DNA or RNA, is made up of many nucleotides joined together in a chain-like fashion. Structure of a nucleotide. Each nucleotide has three components. Nitrogenous base. Two types. Purines. Adenine A. Guanine G. Pyrimidines. Cytosine C. Thymine T in DNA. Uracil U in RNA. Pentose sugar. Ribose in RNA. Deoxyribose in DNA. Phosphate group. From nucleotide to chain. How it happens. Nitrogen base plus pentose sugar forms a nucleoside. E.g., adenosine, guanosine, cytidine, thymidine, uridine. A nitrogen base attaches to pentose sugar. How? Formation of nucleoside. Type of bond. When a nitrogenous base attaches to a pentose sugar, either ribose in RNA or deoxyribose in DNA, the two are linked by a beta-N glycosidic bond. Bond details. Type of bond. Beta-N glycosidic bond. Between 1' carbon of the sugar. Nitrogen atom in the base. End 9 of purines, adenine, guanine. N1 of pyrimidines, cytosine, thymine, uracil. Nucleoside plus phosphate forms a nucleotide. At 5' carbon of pentose sugar, make bond with OH group of phosphate molecule. And there you go. You have just made nucleotide. See video. This is just one nucleotide. When such two molecule join together they forms dinucleotide. See video. Nucleotides join through 3' 5' phosphodiester bonds to form dinucleotide, where 3' carbon of pentose sugar and 5' carbon of phosphate group of another nucleotide. Formation of polynucleotide chain, DNA/RNA. Nucleotides join together through phosphodiester bonds between the 3' OH of one sugar and the 5' phosphate of the next. Reaction. Nucleotide plus nucleotide, polynucleotide, DNA/RNA, via 3' 5' phosphodiester linkage. The result is a sugar-phosphate backbone with attached nitrogenous bases. Ends of the chain. 5' end free phosphate group. 3' end free OH group on sugar. The backbone of DNA/RNA equals sugar plus phosphate. Bases project out from the backbone. When two polynucleotide strands join to form a DNA double helix, they are held together by hydrogen bonds between complementary nitrogenous bases. In the DNA double helix, two polynucleotide strands come together to form a stable, twisted ladder-like structure. But how do they join? The answer lies in the unique hydrogen bonds that form between nitrogenous bases of the two strands. Adenine always pairs with thymine through 2 hydrogen bonds, while guanine pairs with cytosine through 3 hydrogen bonds. This specific pairing is known as complementary base pairing. These hydrogen bonds, though weak individually, collectively provide significant stability to the DNA structure. Additionally, the two strands run in opposite directions, one from 5' to 3', and the other from 3' to 5'. This arrangement is called antiparallel orientation. Together, base pairing and antiparallel arrangement hold the DNA strands tightly and precisely, creating the iconic double helix, the molecule of life. Let's understand the key differences between DNA and RNA, the two fundamental nucleic acids in living organisms. First, sugar type. DNA contains deoxyribose, while RNA contains ribose. Ribose has one extra oxygen atom, giving RNA a reactive edge. Second, nitrogen bases. DNA uses thymine T, but in RNA, thymine is replaced by uracil U. Third, structure. DNA is typically double-stranded, forming a double helix. RNA is usually single-stranded, which allows it to fold into various shapes for different functions. And finally, the extra OH group. DNA lacks a hydroxyl group at the 2' carbon of its sugar, making it more stable. RNA, on the other hand, has this OH group, which makes it more reactive and less stable than DNA. In short, DNA stores genetic information, while RNA plays a dynamic role in expressing it. Discovery of DNA. Friedrich Miescher, 1869, discovered DNA, called it nuclein. Structure remained unknown for decades due to technical limits. The double helix model of DNA, proposed by Watson and Crick in 1953, based on X-ray diffraction data by Rosalind Franklin and Maurice Wilkins. Chargaff's rule. A equals T and G equals C in any double-stranded DNA. Salient features of the double helix. 2 polynucleotide chains. Sugar phosphate forms the backbone. Bases project inward. Antiparallel strands. One strand 5' to 3'. Other strand 3' to 5'. Base pairing via hydrogen bonds. Adenine and thymine has 2 hydrogen bonds. Guanine and cytosine 3 hydrogen bonds. Purine always pairs with pyrimidine uniform helix width. Right-handed helical coil. Helix dimensions. Pitch 1 full turn 3.4 nanometers. 10 base pairs per turn. Distance between adjacent base pairs. 0.34 nanometers. Base stacking. One base pair sits on top of another, contributing to helix stability. The central dogma of molecular biology. Revolutionary impact of the DNA double helix. The double helix structure of DNA proposed by Watson and Crick not only explained how DNA stores genetic information, how it replicates faithfully, but also opened the doors to understanding how genes control cell functions. This discovery marked a turning point in molecular biology. Francis Crick's central dogma. Soon after the discovery of DNA structure, Francis Crick proposed the central dogma of molecular biology. What is the central dogma? The central dogma describes the flow of genetic information within a biological system. DNA to RNA to protein. Step-by-step flow. DNA, deoxyribonucleic acid, stores genetic information. Replicates during cell division. RNA, ribonucleic acid, made from DNA by transcription, acts as a messenger, mRNA, adapter, tRNA, or part of machinery, rRNA. Protein, synthesized from RNA through translation. Proteins perform all structural and functional roles in cells. Why is this important? It explains gene expression. DNA holds the blueprint. RNA is the copying technician. Protein is the final product or worker. This concept revolutionized genetics, cell biology, and biotechnology. 5.1.2 Packaging of DNA helix. Flow of genetic information, from RNA to DNA. Before we jump into packaging, let's answer this. In some viruses, information flows from RNA to DNA. What is this process called? This process is called reverse transcription. It occurs in retroviruses like HIV. Enzyme involved. Reverse transcriptase. Central dogma is reversed. RNA DNA RNA protein. Length of DNA in a cell. Let's do the math. Human DNA. Total base pairs. BP 6.6 times 10. Distance between base pairs. 0.34 nanometers equals 0.34 times 10 m. Total length of DNA equals 6.6 times 10 times 0.34 times 10 equals 2.2 meters. A single human cell's DNA is 2.2 m long, but it fits into a nucleus that is just 1 to 2 m, 10 m, in diameter. What about E. coli? Given length of E. coli DNA equals 1.36 millimeters equals 1.36 times 10 superscript 3m. Base pair distance equals 0.34 times 10 m. Base pairs in E. coli DNA equals 1.36 times 10 superscript 3m / 0.34 times 10 m equals 4 times 10 BP. Approximate. DNA packaging in prokaryotes, e.g. E. coli. No true nucleus, but DNA isn't floating randomly. DNA is negatively charged. It binds to positively charged proteins. Forms a dense region called nucleoid. DNA is looped and compacted by loop-forming proteins. DNA packaging in eukaryotes. Much more complex and organized. Key players. 1. Histones. Small positively charged proteins. Rich in lysine and arginine. These amino acids have positive side chains. 2. Histone octamer. 8 histone proteins form an octamer. DNA, which is negative, wraps around this protein spool. 1 unit equals nucleosome. Each nucleosome is almost equal to 200 base pairs of DNA. Visual. Think of thread DNA, wrapped around beads, histones. Chromatin equals DNA plus histone proteins. When viewed under an electron microscope, chromatin looks like beads on a string. Each bead equals nucleosome. String between beads equals linker DNA. Higher-level DNA packaging. Nucleosomes, chromatin fiber. Chromatin supercoiled chromosomes. Especially visible during metaphase of cell division. Additional proteins. Non-histone chromosomal NHC. Proteins assist in further folding and compaction. Types of chromatin. Chromatin type packing appearance function. Euchromatin loosely packed light stain transcriptionally active. Heterochromatin densely packed dark stain inactive. Silent DNA. 5.2 The search for genetic material. Stepwise explanation. Step 1. Initial discoveries. Mendel and Miescher 1800s. Gregor Mendel 1865. Discovered basic principles of inheritance using pea plants. He talked about factors responsible for traits, what we now call genes. Friedrich Miescher 1869. Discovered a mysterious substance in the nucleus, rich in phosphorus. He called it nuclein, which we now know is DNA. Step 2. Doubt and confusion, late 1800s. Early 1900s. Even though both inheritance and DNA were known by then, scientists did not yet know that DNA was the genetic material. The question still remained. Which molecule carries the instructions of life? DNA, RNA, or proteins? Step 3. Chromosomes come into focus, early 1900s. Walter Sutton, 1902, proposed the chromosomal theory of inheritance, linking chromosomes with heredity. Thomas Hunt Morgan, using fruit flies, Drosophila. He demonstrated that genes are located on chromosomes and discovered linkage and gene mapping. But chromosomes contain both proteins and nucleic acids. So, which one carries the actual genetic instructions? Step 4. Turning point. Molecular level analysis. By 1926, scientists began focusing on the molecular nature of genes. They now knew genes were part of chromosomes. Chromosomes were made of proteins plus DNA. But which one was responsible for transferring traits? So, research now focused on proving experimentally whether it was DNA, RNA, or protein that stored genetic information. Conclusion of the step. By the end of this period, scientists knew chromosomes carry genes. We're still debating the chemical nature of the genetic material. Laid the foundation for later experiments like Griffith's, Avery McLeod McCarty, and Hershey Chase. Transforming principle. Griffith's experiment, 1928. Background. Frederick Griffith, a British bacteriologist, was studying Streptococcus pneumoniae, a bacterium that causes pneumonia. He worked with 2 strains. S-type, smooth. Virulent, causes disease. Has a polysaccharide capsule. R-type, rough. Non-virulent, harmless. No capsule. Let's understand the classic experiment by Frederick Griffith, which led to the discovery of the transforming principle, a major step toward identifying DNA as the genetic material. Experiment 1. Live S strain. In the first trial, Griffith injected live S strain bacteria into healthy mice. These bacteria had a smooth capsule and were virulent. The result? The mice died. This confirmed that the S strain was disease-causing. Experiment 2. Live R strain. Next, he injected live R strain bacteria, which lacked the capsule and were non-virulent. This time the mice survived. So, the R strain alone was harmless. Experiment 3. Heat-killed S strain. In the third experiment, Griffith used heat-killed S strain and injected it into mice. Since the bacteria were dead, the mice again survived. This showed that dead S strain bacteria alone could not cause disease. Experiment 4. Heat-killed S strain plus live R strain. Then came the surprising part. Griffith mixed heat-killed S strain with live R strain and injected the mixture into mice. To everyone's shock, the mice died. Even more surprisingly, live S-type bacteria were recovered from the dead mice. Conclusion. Griffith concluded that some transforming principle from the dead S strain had converted the R strain into virulent S-type. He didn't know it then, but this transforming principle was DNA. Griffith's conclusion. Something from the dead S strain was transferred to the live R strain, making it virulent. This mysterious substance transformed the R strain into an S strain. He called it the transforming principle. But what was the transforming principle? Griffith did not know whether the transforming principle was DNA, RNA, or protein. Later, Avery, MacLeod, and McCarty, 1944, discovered that it was DNA that caused the transformation, proving that DNA is the genetic material. Avery MacLeod McCarty experiment, 1944. Detailed pointwise explanation. 1. Background and aim. In 1928, Griffith discovered a transforming principle that turned non-virulent R strain into virulent S strain. However, he didn't identify what the transforming substance was. Avery, MacLeod, and McCarty set out to find what molecule, DNA, RNA, or protein, was responsible for this transformation. 2. Source material used. They used heat-killed S strain bacteria, just like Griffith. They extracted different biomolecules, DNA, RNA, proteins, etc., from the S strain. 3. Experimental setup. They divided the extract into separate test tubes and treated each with different enzymes. A. Protease treatment. Destroys proteins. S strain extract plus protease enzyme. Destroys proteins. Then mixed with live R strain bacteria. Result. Transformation still occurred. R strain became virulent S strain. Conclusion. Proteins are not the transforming principle. B. RNase treatment. Destroys RNA. S strain extract plus RNase enzyme. Destroys RNA. Then mixed with live R strain bacteria. Result. Transformation still occurred. R strain became virulent. Conclusion. RNA is not the transforming principle. C. DNase treatment. Destroys DNA. S strain extract plus DNase enzyme. Destroys DNA. Then mixed with live R strain bacteria. Result. No transformation occurred. R strain stayed non-virulent. Conclusion. DNA is essential for transformation. 4. Final conclusion. Only DNase destroyed the transforming ability, proving DNA is the transforming principle. DNA carries genetic information. 5. Importance of the experiment. First, clear evidence that DNA is the genetic material, not protein. A turning point in molecular biology. Laid foundation for Hershey-Chase experiment, which confirmed DNA's role using viruses. Summary table. Enzyme used, molecule destroyed, result, conclusion. Protease, protein transformation, protein not genetic material. RNase, RNA transformation, RNA not genetic material. DNase, DNA no transformation, DNA is genetic material. Hershey-Chase experiment, 1952. Alfred Hershey and Martha Chase. 1. Background. By 1950s, it was still debated whether DNA or proteins carried hereditary information. Hershey and Chase used a virus that infects bacteria, called a bacteriophage, specifically, T2 phage. Bacteriophage equals a virus with a DNA core and a protein coat. 2. Objective. To determine whether DNA or protein from the virus enters the bacteria and carries genetic information. 3. Method and materials. They used radioactive isotopes to label DNA with radioactive phosphorus, 32P. Since DNA contains phosphorus, proteins do not. Protein with radioactive sulfur, 35S. Since proteins contain sulfur, DNA does not. 4. Steps of the experiment. Step 1. Labeling the phage. Grew phages in 2 different media. 32P labeled DNA. 35S labeled protein coat. Step 2. Infection. Let both types of labeled phages infect E. coli bacteria. Step 3. Blending. After infection, they blended the mixture to shake off viral coats from bacterial cells. Step 4. Centrifugation. The mixture was centrifuged to separate heavier bacterial cells, pellet, lighter viral parts, supernatant. 5. Observations. Phage label where the radioactivity went, conclusion. 32P DNA. Inside the bacterial pellet, DNA entered cells. 35S protein, in the supernatant, outside cells. Protein stayed outside. 6. Conclusion. Only DNA enters the bacteria and directs viral replication. Hence, DNA is the genetic material, not protein. 7. Significance. Final proof that DNA, not protein, is the molecule of heredity. Strong support for Avery-MacLeod-McCarty findings. 5.2.2 Properties of genetic material. DNA versus RNA. Background. The Hershey-Chase experiment confirmed DNA as the primary genetic material in most organisms. However, some viruses like TMV and bacteriophage QB use RNA as their genetic material. Why DNA is preferred as genetic material over RNA. Criteria for a molecule to be genetic material. Requirement explanation. 1. Replication should be able to make exact copies of itself. 2. Stability should remain unchanged during life cycle or environmental changes. 3. Mutation must allow slow changes for evolution. 4. Expression should express traits, Mendelian characters. Comparison between DNA and RNA. Feature 1. Sugar. DNA contains deoxyribose, which lacks an hydroxy group at the 2' position, making it more stable. RNA contains ribose, which has an hydroxy group at 2', making it more reactive. Feature 2. Nitrogen bases. DNA uses adenine, thymine, guanine, and cytosine. RNA replaces thymine with uracil, so its bases are adenine, uracil, guanine, and cytosine. Feature 3. Strand type. DNA is usually double-stranded, forming the famous double helix. RNA is usually single-stranded, allowing more varied structures. Feature 4. Stability. DNA is chemically stable, thanks to the absence of the 2-OH group and presence of thymine. RNA is less stable, due to its reactive 2-OH group. Feature 5. Reactivity. DNA is relatively inert. RNA is more reactive and even has catalytic roles, like ribozymes. Feature 6. Mutation rate. DNA has a slow mutation rate, ideal for storing long-term genetic information. RNA, especially in viruses, has a higher mutation rate, helping them evolve quickly. Feature 7. Role in protein synthesis. DNA's role is indirect. It stores information but requires RNA to make proteins. RNA plays a direct role as messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA, rRNA. Key concepts. DNA is structurally and chemically more stable due to lack of 2' OH group, presence of thymine instead of uracil in RNA. RNA is catalytic and reactive, useful in expression and enzymatic functions, more prone to mutations, faster evolution in RNA viruses. DNA is a better long-term storage molecule, while RNA is ideal for short-term expression. Can you recall 2 chemical differences between DNA and RNA? Sugar in DNA, deoxyribose in RNA ribose, nitrogen base, thymine T in DNA is replaced by uracil U in RNA. Conclusion. DNA is better suited as the genetic material due to its higher stability and lower reactivity. RNA can also act as genetic material in some viruses, and plays a crucial role in gene expression and protein synthesis. Extra exam questions. Assertion. Reason. Assertion. DNA is more stable than RNA. Reason. DNA lacks the 2' OH group that makes RNA more reactive. Answer. Both assertion and reason are true, and reason is the correct explanation. MCQ. Which of the following is not a feature of ideal genetic material? A. Ability to replicate. B. High instability. C. Ability to mutate. D. Ability to express characters. White heavy checkmark. Answer. B. High instability.